Welcome back after the lunch break. I hope you had a great lunch. We have a slight change of the program this afternoon. Since Felix Shortman has not arrived yet, we will start with George Mashour. George is one of the people that really run this conference for a few years now, and he's from the Center for Consciousness Science at the University of Michigan at Ann Arbor. Welcome with me, George Mashour.
It's really a great pleasure to be here, and I'd like to thank Harold and Stuart for the kind invitation to speak. Before I start my lecture, I have some preliminary but nonetheless important comments to make. First of all, I am not a quantum physicist, and I'm not going to pretend like I'm a quantum physicist. I'm a neuroscientist who studies connectivity and graph theoretical variables in both animals and humans who are going in and out of states of unconsciousness, primarily general anesthesia, but also sleep and pathological disorders of consciousness. And I'm also a clinical anesthesiologist. So that begs the question, why am I here speaking in a plenary session on quantum physics as opposed to neuroscience, which is my usual home at the Tucson meetings and elsewhere? Well, I have to admit, I don't have a particular affinity for quantum theories of consciousness. I don't see a compelling reason to invoke quantum physics at this stage of investigation. I believe primarily in a neuroscientific pathway to understanding consciousness, and I suspect that neuroscience will have decades, if not centuries, before it needs to invoke quantum physics. But on the other hand, I'm also not repelled by the theory. I'm not antagonistic toward it, and that's because I try to keep an open mind. I feel like we know very little about consciousness, and it's important to consider this theory. And I think the theory is of particular importance to the science of consciousness in general, and that's why I have been getting more directly engaged with it, and I think others need to get engaged with it. I think it's important because if the theory is proven to be true, it's really going to be a massive discovery, and not just massive for the science of consciousness, massive for science in general, more so than other theories of consciousness if they were proven true. On the other hand, if the theory is not true, it really constitutes a major distraction for the field, and because of some of the New Age implications that are ascribed to the theory, not by the theorists themselves per se, it also has somewhat compromised or brought into question the scientific legitimacy of the field of consciousness science. And so I think it's very important to address this, and the stakes are quite high, I think. Now, I also believe that it's time to start taking an empirical or experimental approach to this theory. I think that's where the critical progress is going to be made in terms of understanding whether we progress along this line, acknowledging that it's high risk but high reward, but at least plausible, or whether or not we need to course correct and exclude that as a methodology for exploring consciousness. And that's the spirit in which I've approached these early experiments. Now, I have to admit, the thought of trying to empirically address quantum theories of consciousness was overwhelming, but I thought a more tractable approach would be to explore a corollary theory or a dimension of the theory, and that is the mechanism of anesthetic-induced unconsciousness, which in the Penrose-Hameroff framework has a quantum mechanism. Then I could move away from a network that has billions of nodes and trillions of edges to a single anesthetic molecule. And I can also feel confident about studying anesthetic molecules because they work 100% of the time. There has never been a single case since the beginning of the field of anesthesiology in the mid-19th century of an individual that could not be anesthetized. And furthermore, not only do they work across all humans that have ever been tested, and that's unique in terms of medicine. There's no other class of drugs that works 100% of the time. They also work across all of biology. Other mammalian species, Drosophila, the fruit fly, C. elegans, the worm, plants, and even single-cell organisms. And this touches on some of the earlier discussions this morning. So there's a molecule or a set of molecules that works 100% of the time in humans to more or less selectively ablate consciousness and also works across all of biology. So that's the experimental framework. And my last preliminary comment is that these experiments are preliminary. This is a first step, but I think a critical step. So I'm going to start by just contextualizing the problem of anesthetic mechanisms for you. I'm going to touch ever so briefly on quantum theories of consciousness and anesthesia. You've heard from Roger Penrose. You're going to hear from Stuart Hameroff. You don't really need to hear from me, but I will touch on some key points. And then also I'm going to describe what I consider the first experiments testing directly a quantum theory of anesthetic mechanism with both classical and entangled photons. And I found it important to try to get at the heart of quantum theories of consciousness by engaging, or entangling, if you will, with entanglement to address, at least in an experimental model, this notion that somehow entanglement or non-local processes mediate the binding of information in the brain that gets disrupted during general anesthesia. So first, a historical framework to help put into context what I'm working on and what Stuart Hameroff will no doubt talk about soon. The first public demonstration of general anesthesia happened October 16th, 1846 at the Massachusetts General Hospital of Harvard Medical School. And that's actually where I trained as a clinical anesthesiologist. And this was revolutionary because at the time, surgery and pain were just considered to be inextricably linked. The concept of a painless surgery was almost fantastical at the time. And this is the first major American contribution to medicine, and the word spread worldwide. And around that time, ether, which was used here, was employed as an anesthetic, but also chloroform and nitrous oxide. And very soon, people started to ask the question, how is it that these three structurally and chemically, and now we know neuroscientifically diverse molecules can all achieve what appears to be the same functional outcome of unconsciousness? So this happened in October. The first report and the precursor to the New England Journal of Medicine was published in November. And within a year, von Bibra and Harless put forward the first theory of anesthetic mechanism, which is that anesthetics dissolved lipids in the brain. Now, they didn't explain how those lipids reconstituted and we woke up at the end, but that was the first theoretical framework. And then we get to the 1860s and Claude Bernard, who made several fundamental contributions. First of all, Bernard explored anesthetic effects in a number of different species, including plants, which is going to be relevant to my talk later. He also proposed what is now referred to as the unitary hypothesis of anesthetic mechanism. That is, that there's some kind of final common substrate or mediator that accounts for the fact that all of these diverse agents converge on a single functional outcome. But nobody really knew what the biological substrate of that was until the early 1900s, when Hans Meyer and Chester Overton independently discovered that the potency of an anesthetic is directly variable with its solubility in olive oil. So the more lipophilic the anesthetic, the more potent the anesthetic. And that established what is referred to as the Meyer-Overton relationship. And it was thought that anesthetics non-specifically affected membrane fluidity in the bilipid membrane of neurons. And this paradigm for anesthetic mechanism persisted throughout most of the 20th century, until in the 1980s, Nick Franks, who's still very active in this field, and Matt Lieb discovered that anesthetics could function in a Meyer-Overton relationship in a lipid-free environment. And in fact, they could directly modulate the function of a protein. In this case, it was firefly luciferase. And that opened up many different lines of investigations because now things like neurotransmitter receptors and ion channels could be explored and investigated in terms of anesthetic binding. And in fact, In the next decade, and this is Neil Harrison, who is the first to identify a binding site for general anesthetics on the GABA receptor. GABA being gamma-aminobutyric acid, the primary inhibitory neurotransmitter in the brain. Anesthetics were thought to potentiate GABAergic transmission and inhibit neural function. And then when we move into the 2000s, and this is where I've been active, I started as a faculty in 2007, this is really the era of networks, thinking about sleep-wake networks, thinking about functional, directed, and effective connectivity across anesthetic state transitions, and also, and this is something that my laboratory has really focused on, applying graph theory and network science to anesthetic state transitions. But all of this presupposes a classical perspective of the brain. And of course, as we all know here, and as you've heard, there are alternative perspectives. One primary theory and prominent theory introduced in the late 1980s, and again, you've heard from Professor Penrose, so I don't need to recapitulate it here, but essentially proposing something that was non-algorithmic, non-classical, and possibly requiring quantum physics. But the biological substrate for that in the brain was unknown until Stuart Hameroff basically connected with this theory. He was working on microtubules, you'll hear more about them, but suggesting that perhaps the lattice of microtubules throughout the brain functioned as the biological substrate that could mediate the kind of quantum events required for conscious experience. And part of the reason why Stuart was investigating microtubules had to do with single-cell organisms. They can be attracted to things, they could be repelled. As Stuart liked to say, they can find a mate, they can have sex, they can do a lot of things, but they don't have a nervous system. In fact, they don't even have a neuron. So the question arises, what is the nervous system, what is the information processing system of a single-cell organism? And Stuart pointed to cytoskeletal elements, and in particular, microtubules, as the basis for that information processing. I'm sure that you will be seeing this, or at least hearing about it, in Stuart's talk, so I won't go into it, but you can basically see the different scales of the neuronal dendrite to the microtubule to the tubulin, and the possible quantum effects that are happening within those microtubules. Now, I wanted to note that anesthetic effects or anesthetics potentially working through microtubules is not a new idea. It's not original to Stuart. And Stuart, of course, gives credit to Allison and Nunn, who in the late 1960s, in this Lancet article, suggested that anesthetics worked on microtubules. And I point that out for historical significance. They came to the same conclusion because of anesthetic effects in single-cell organisms and observed alterations of microtubules. But I do want to point out that there are some reasons to believe that anesthetics might work through microtubules that are independent of quantum theory. Also, microtubules are abundant, and general anesthetics have very promiscuous binding patterns. And so it's not unexpected that microtubules would be a target for general anesthetics. Now, what is the current evidence related to any kind of connection between anesthetic mechanism and quantum physics? It really comes down to two articles. There's some modeling studies that are suggestive and that Stuart will discuss, but in terms of some empirical evidence of even connecting anesthetic effects with that kind of scale, it comes from two articles published in prominent journals, one in PNAS, looking at electron spin changes during general anesthesia in Drosophila, that's the fruit fly. They go into a state of behavioral quiescence in the presence of anesthetics. But this was done at very, very, very cool temperatures. There was also a more recent paper, and Stuart wrote an editorial on it, showing that nuclear spin is related to anesthetic potency. And that was using xenon isotopes. So xenon, as you all know it, a Nobel gas, it's inert. It's actually one of the best anesthetics out there. The problem is it's about $20,000 a tank, so it's not used, but it has a very fast onset, a very fast offset, and it's an excellent anesthetic. And this demonstrated that the isotopes really have a different anesthetic potency. But there's been no direct assessment of whether or not a general anesthetic molecule can have a quantum interaction. And that is what I wanted to address. And again, wanting to try to get at the heart of the matter by thinking about a system in which we might explore anesthetic effects on entangled particles, as first predicted by Einstein, Podolsky, and Rosen, and as has been demonstrated for electrons and photons, and at really remarkable distances. So this cover article from Science in 2017 shows entangled photons that are essentially getting shot out from space, and that demonstrate entanglement at over 600 miles apart on the surface of China. So, very well documented. Now, my quest, and this is something that's been bubbling in the back of my mind, again, trying to figure out how can we get some experimental traction on some dimension of this theory. My quest was to find some kind of model system that had a macroscopic quantum property, so I didn't really need to get down to a subatomic level. And there are examples of this, and for instance, Bose-Einstein condensates. So it had to have some kind of macroscopic quantum property. It has to exhibit such properties at biologically relevant temperatures. Because one of the big critiques of OR is that this kind of orchestrated reduction can't happen in the warm, wet, noisy environment of the brain. So this quantum system needs to be functional at hotter temperatures. And finally, there has to be some kind of a priori evidence to suggest that this system is susceptible to the effects of general anesthetics. Otherwise, I could be spending years just trying to empirically test different systems. So there had to be some pretest probability that the system would be subject or susceptible to the effects of general anesthetics. And so after reflecting on this, and maybe you all have already come up with a model, but it took me a long time as this was brewing in the back of my head while I was going about my neuroscientific work, that photosynthesis, or something related to photosynthesis, would actually be an excellent model in which to test general anesthetics, because photosynthesis does have quantum properties that could be observed and measured involving entangled photons. In fact, photosynthesis is often put forward as a canonical example of quantum biology and the relevance of quantum physics for biology. We know it's going on at hot temperatures. It's going on right now in hot Interlaken, right outside the door. And Claude Bernard, who I put up on the screen from the 1860s, actually demonstrated that anesthetics inhibited photosynthesis. So this seemed like a really good approach, something related to photosynthesis or the chromophores that are mediating photosynthesis might be relevant. So then I had to spend another couple years trying to figure out how I was going to test this. Sent many emails to many people who never responded because they probably thought I was crazy as an anesthesiologist to want to be testing molecules in some kind of quantum system. But then I was connected, actually out independently, with Ted Goodson, who is a professor of chemistry and a professor of applied physics at the University of Michigan. We met, we talked, I went through my spiel that I just went through with you, and after years of me having dead ends, he said, "Oh yeah, I do that in my lab." I said, "What?" So as it turns out, he's really interested in the way both natural and synthetic chromophores handle quantum light. And so I said, take me through this. And as it turns out, there is a reproducible way of generating entangled photons. And the process is called spontaneous parametric downconversion. But essentially what happens is you shoot in a laser on one end, it goes through a crystal, and out come entangled photons. And this is something that he does empirically in his laser lab. And this is what it looks like schematically. And so he was describing the process of this laser going through, in this case, barium borate crystal, generating entangled photons. And I said, well, what are you measuring? What is it going through? And ultimately, it passes through a sample. There's a little plastic cuvette. There's an organic solvent. And there is some kind of chromophore that he tests. Again, either natural or synthetic. And then you have a photodiode over here that is registering the photons that are hitting it. And it essentially takes advantage of the photoelectric effect, except the photons are going in and the electricity is coming out. And so I said, well, could we put an anesthetic in this cuvette, either with a chromophore to test some kind of interaction or on its own? And so after going down a couple of more dead ends, I decided, just as a proof of principle experiment, just to see if we could technically get through this, to study two modern anesthetic ethers. And these are used in operating rooms throughout the world, sevoflurane and isoflurane. Now, normally this would be vaporized and given to you in a gas form at maybe 1% for isoflurane or 2% for sevoflurane of the total gas flow. But in this case, because this required an organic solvent, I thought, why don't we just put in the pure liquid phase ether? It's organic. It's going to mix with whatever the solvent is and test this. And now I'm going to just give you one data slide that helps explain these effects. So first of all, I want to direct your attention to up here, to this part of the slide on the top, where this is classical two-photon excited fluorescence. So it's generating photons, but they're not entangled photons. And what you can see as you increase the photon flux, this is a positive control, coumarin 153. You can see an increase here. But if you look at the pure sevoflurane, the pure isoflurane, or the solvent, methanol, nothing happens. Okay? So in other words, there doesn't appear to be any interaction between isoflurane or sevoflurane, these anesthetic ethers, and classical light. And what you're seeing down here, and notice that these are orders of magnitude higher than the photon flux here. Now you're looking at the entangled photon absorption. All right, so the laser is going into the crystal, it's generating entangled photons, it's going through the sample, it's transmitting to the photodiode, and then you can make an inference about what was absorbed by that molecule. Either the chromophore, which is the positive control, and you can see that as the photon flux increases, you start to get the higher absorption, or the negative control, which is the methanol solvent. But look at where isoflurane and sevoflurane fall on this logarithmic y-axis. They actually are significantly elevated in terms of the absorption of the entangled photons compared to the negative control. So what you're seeing here is that these molecules, these anesthetic molecules, are essentially transparent when it comes to classical light, but they appear to absorb entangled photons. Now, my colleagues have done this work. They were very rigorous about it. They tested the agents with mass spectroscopy to ensure that there was high purity. They did other kinds of modeling to try to understand the mechanism. This was replicated. It was reproduced. Now, this doesn't mean that you could put any kind of molecule in here and get the same result. And in fact, the more classical ethers, so diethyl ether or methyl isopropyl ether, had the opposite kind of effect, where they absorb the classical light, but they don't seem to interact with these entangled photons. The difference between those earlier ethers and these modern ethers is a process called halogenation, and that is essentially putting fluoride around, how much time? Okay, great. Fluoride on the molecule to reduce its volatility. Diethyl ether was a great anesthetic. It also made operating rooms explode. And so they had to move to less flammable agents. So I want to summarize. The electronic state of modern anesthetic ethers that we're currently using directly and selectively interact with entangled photons. The biological significance is unknown, and I don't want to make any conclusions as to whether or not photons running through optical channels and a microtubule lattice are interrupted by anesthetic ethers. It's pure ether, it's in a liquid form, so we don't know what to do with it biologically. But this system could be modified to become more biologically relevant and maybe to model some other things that we might be interested in. And the reason I'm excited about it is, for me at least, it is a first step experimentally with a rigorous approach to empirically demonstrate what is really the first question that needs to be asked for this particular aspect of quantum theories. Namely, can an anesthetic have a direct interaction with some kind of quantum system? And in this case, the answer is yes. So I want to thank again Ted Goodson. These are some of his colleagues. That's my colleague, Tony Huditz. I want to thank all of my neuroscience colleagues at the Center for Consciousness Science, which I founded and direct at the University of Michigan Medical School. And I thank you for your attention, and I look forward to see what bubbles out of the quantum foam during the discussion later. Thank you.
Thank you. So, is it on? Yes. Thank you so much, George. That was a great talk, fascinating results, and encouraging results. Next speaker will be Felix Shortman. I'm particularly happy to welcome Felix because he is from the same place where I am. I'm ETH, but he's University of Zurich, and I think also ETH. Is that right?
University of Zurich.
University of Zurich. Felix will talk about the role of light in understanding brain function. Thanks for coming, Felix.
Thank you very much for the introduction, and thank you also for inviting me and having the opportunity to give a summary about a quite recently defined research area termed neurophotonics. Neurophotonics, as I am defining it, comprises 3 different categories of research. The first is optical neuroimaging. Second is cellular and tissue autoluminescence. And the third one is light effects on brain tissue and function. So combining these 3 areas together gives the research area of neurophotonics. It's an interdisciplinary field involving biophysics, physics, engineering, cell biology, human physiology, pathophysiology, and of course neuroscience. Let's start with the first block, optical neuroimaging. It was in 1977 where in Science was a very interesting paper published from Professor Franz Jöpsis. He demonstrated that it is possible to shine light from the one hand side into the head and detect the emitted light with a very sensitive photomultiplier tube and infer some physiological information from this setup. This was quite remarkable because the amount of light detected is extremely low. The next thing he was showing was that when he asked the subject to change the respiration rate, then there was an increased amount of light detected. And this was an indication that he was measuring actually the blood volume in the head with his setup. So he concluded near-infrared light can penetrate the whole head and the brain, and the absorbed near-infrared light is an indicator for brain blood perfusion. And this was then the conclusion that there's a new technique that needs to be discovered and implemented based on this discovery, that is able to measure cerebral blood perfusion non-invasively in humans. The next step in the story was to not only measure with one wavelength, but with multiple wavelengths. And why? Because if you measure the color of the blood, then it's able to infer the oxygenation state of the blood, which is a very critical physiological parameter. So now we know that the absorption spectrum of blood depends heavily on the oxygenation state of hemoglobin. And using this knowledge, it's possible to develop technology that can differentiate between the oxygenated and deoxygenated form of hemoglobin, oxyhemoglobin and deoxyhemoglobin. So the first step was the next development using multiple wavelengths. And then another thing happened. It was also shown that when placing detectors at several positions, it's possible to infer additional information than only one channel. So this multi-light path approach appeared. And now we are in a state where both approaches are combined. And we have actually two main technologies, functional near-infrared spectroscopy and near-infrared spectroscopy-based cerebral oximetry. This is a slide showing the devices that are on the market and that were developed in the last couple of years. These are the most famous persons, I would say, in the moment in this field: Professor Chance, Boas, Elwell, and Professor Wolf. Professor Wolf is from the University of Zurich and is actually my supervisor, of a PhD supervisor and the head of the lab where I'm working. And this is one of the newest developments. It's really a small sensor that you can place on the head and is performing these optical spectroscopy measurements. We also developed sensors that can be used in non-standard populations like preterm-born children to measure what is going on in the brain. And these are the more advanced concepts. As you can see, it's still a challenge to place all these optical elements on the head. This is an interesting configuration, but also quite nice because it combines EEG and optical spectroscopy. So you have two modalities linked together. This is a more classical diffuse optical spectroscopy system measuring it with many channels simultaneously. Okay, so what can be measured? Two things. The absolute value of a concentration, for example here, the tissue oxygenation. And the other thing is to look how the concentration changes with time when there was a stimulation, for example, like a cognitive task or brain activity. Okay, the bigger picture is the following. Neural activity can be measured, of course, by looking at the electrophysiology and the magnetic field properties with EEG and MEG. Then, of course, there's also mechanical and electromagnetic field properties changing with neural activity that can be measured also, not really in a clinical context, but in an experimental context. And then we have blood flow and tissue metabolism, which is measured normally with fMRI, but now it can be measured also with the optical methods. What are we measuring? We are measuring actually the blood flow and oxygenation in the superficial part of the brain, the cerebral cortex, which is quite small, so about 1.5 to 4.5 millimeters. And in this specific region, there's a lot of happening during neural activity. We have the PL, venular, and arterial network, some penetrating arterioles and venules. And in this tissue bed, there's an interesting process happening. It's called neurovascular coupling. And it's basically a process linking brain activity to changes in blood flow and tissue oxygenation. It's a very complicated process. I won't go into details about it. And interestingly, it's also not completely understood what is actually happening. So there are still things to discover, why this is happening and how it's happening. But we can measure it. This is a study I finished 1 year ago, measuring these changes happening in the occipital part of the head during visual stimulation in newborns. This is a small child. I have here a LCD monitor showing quite strong light flashes and detecting what is going on in the visual cortex, and was nicely visible that there's an increase in blood flow. This is the newest study. It's also actually the first study investigating effects of psychedelics with NIRS. It's just a single subject study to see if it's possible, and what we found is there is a change in functional connectivity, which can be also measured with FNIRS happening during the states. This was 30 minutes after the administration of 70 milligram of psilocybin, and it's 60 minutes. So this is the first result. Another application is using this technique in clinics. The relevance is here highlighted. During an operation, there's normally only the blood oxygenation in the finger method measured, the arterial oxygenation, but not the oxygenation in the brain. But there are studies that did this, and they found, there's a drop in oxygenation, quite a large drop during the operation, and this could be linked to a problem, postoperative cognitive dysfunction that the subjects were experiencing. Interestingly, it could be shown that it's possible to predict the occurrence of POCD based on the drops in cerebral oxygenation during the operation. So it's an immediate medical application. It's still not routinely used in the clinics, but I think in the next couple of years it will be. Coming to the second topic, cellular and tissue autoluminescence, also a very fascinating thing. We all emit light and electromagnetic energy, electromagnetic fields. The most prominent part is in the infrared region and can be measured with thermal imaging. But there's another part of the spectrum in the more or less optical region, which is very low, but still it is possible to measure it. And this is a non-thermal radiation. So we are emitting also constantly light in the optical region, in addition to the infrared light being perceived as heat. The first nice imaging study was done by a Japanese group who could show that on the hand it's possible to detect this light, and the amount of light changes depending on the state of the subject. So when the subject was awake, there was a higher emission compared to the state where he was sleeping. And this was a first indication that first, it is possible to measure it, and second, it has a physiological correlation. I did similar studies on myself. I stopped the blood flow on my arm for 10 minutes and looked what is happening in my tissue, and therefore I needed a very sensitive device to detect this small amount of light. I used a photomultiplier that intensifies the amount of signal a million times. And with this setup, I could show that during the occlusion, there's a drop in the light emission of the tissue. And fantastically also, during the phase afterwards, there was a reduced metabolic state and also a reduced emission. So this shows also nicely that the photon emission is linked to metabolic activity. This is the newest study we published. These are images from hands from different subjects taken at different times of the day. And you can see that the amount of light emitted from the hands also varies with chronobiological parameters and depends also on the subject. So this is an ongoing research, and it's interesting to see how dynamic this phenomena is. The first investigation of the human body was also done by a Japanese group, published 10 years ago, and they could show with a very sensitive device and with an exposure duration of 20 minutes to generate one image that the human body emits also light. There's autoluminescence happening. This is the non-thermal emission of the human compared to the thermal emission that can be captured with thermal imaging. There's another group doing this kind of research, very prominent, Roland van Week and Eduard van Week, and they performed similar experiments and investigations that they could show that also the fluctuation of these signals from the body is specific to the body region and the chronobiological state. For example, the abdomen has a weak emission, is relatively constant, compared to the head and the hands, which have a higher emission and more fluctuations and oscillations. Okay, coming back to the terminology, the best technical term is ultra-weak photon emission for this phenomena, and then there are different other terms used in the literature. I prefer to use autoluminescence because it's a more broader term actually, and there's also low-level chemiluminescence and biophoton emission and biophotons. But it's actually referring to the same phenomena. So what is causing it? This is also still investigated. I would say it's a combination of chemical and physical processes, quite a variety, but mainly it involves reactive oxygen species that when they transit from a higher energetic state to a lower state, they emit photons. For example, singulate oxygen has the capability to emit at 3 different wavelengths. And they have also the ability to interact with other molecules. Then the molecules get in another energetically excited state, and then they emit. So there's a secondary reaction also always happening, and there are a lot of molecules in all biological systems that are able to generate light when they are getting excited energetically. For example, I would say this is very interesting, tryptophan, collagen. We have a lot of collagen in our tissue. And one source of the ROS is, of course, the mitochondria. Mitochondria, in the mitochondria, there are several processes happening, and one process was figured out by Jack Tuchinski. He could show that the ion channel activity in combination with the cytochrome C oxidase is able to produce light emission in the infrared region. So this is, I think, quite interesting phenomena and important phenomena. But we have also the excitation of tryptophan, which causes then fluorescence phenomena and radiation in the UV range. So very different spectral range. Mitochondria are important for different reasons, but also for biophysical and biophotonical reasons. Normal, the picture of mitochondria is like this you see in the textbooks, but I want to stress out this is really not the living state of the mitochondria. The mitochondria, when you look in the microscope, is a long filamentous network, and this is more or less an artifact caused by the staining. And we have to be aware that the actual mitochondrial network is very complicated and has this tube-like structure. And this is also important because the biophysics of the mitochondria is very specific and special, and it has been shown in 2004 by Tar and Kühl in a very prominent and famous paper that they have specific optical properties. And it seems that mitochondria can guide light and even have a medium inside that acts like a laser medium, so it can stimulate the emission and can create coherent states. So very interesting. I published a paper summarizing all the experimental results, and I concluded that mitochondria act like electrical cables. There are many phenomena happening inside, on the membrane, and outside of the mitochondria with respect to electrophysiology, but also optics. So maybe we have really optical and electrical cables in the cell. This is the next step in this reasoning. The microtubules are part of the mitochondria, are part of the axon aligned to the mitochondria, and very good work was done by Jack Tuchinski and Professor Craddock, who could show that there's a tryptophan network in the microtubules, and tryptophan is a chromophore that can be excited and has also the ability to release photons. So the whole tryptophan network can act as a specific light guiding network, creating superradiance. Superradiance is a phenomena between classical light and laser light. Very interesting. There's another phenomena related to UPE, that's the phosphane phenomena. So maybe you know it, when you stand up quite quickly, you can see some light flashes. And according to our reasoning, this is actually ultraviolet photon emission. You are seeing with your mind. So you are actually seeing the photons released by different processes. This is still a thing we investigate in more detail. There was also a study done looking if this is possible. They concluded, yeah, we can detect these photons from the eye, but the amount of light detected is not sufficient to trigger the photoreceptor activity. But we think it was more or less a technical problem to detect the light, and it's not ruling out completely our idea. Then, of course, the brain is important in all these discussions. It has been shown that the brain itself, the brain tissue itself, is emitting light. Shown by Kobayashi here. These are brain slide or parts of the brain from a rat. And there was a fluctuation during the experiment, and he could show that the delta wave EEG activity was correlated with the photo emission, which is interesting because it tells you there's a correlation between electrophysiology and this photo emission. Along this line, in the axon, there's maybe also something happening that is not so normally appreciated. It is currently investigated by Professor Simon, who concluded from his simulations that the axon might be able to guide light. So the axon is maybe a light guide, and the light is coming from the photon emission from the mitochondria inside the cell. Very interesting concept. Of course, we have a little bit of problem here. The nodes of Ranvier, there's a specific optical property here in this region, which normally prevents the guidance of light. However, it has been shown that the ion channels here are in a configuration that maybe they act as photoemission elements that enable the propagation of light indeed through the whole axon. There are other research results shown that the brain tissue is specific. For example, the work of Professor Japai Dai. He applied different neurotransmitters to slices of brain tissue, and he could show that, for example, glutamate is increasing the photon emission of the tissue. Then he used different combinations of neurotransmitters, for example glutamate and acetyl-CoA, GABA, serotonin, and also propofol. And he could show that when applying propofol and glutamate, the propofol is reducing the photon emission. How is quantum physics involved in all this? Of course, when doing the simulations and the interpretation of the experiments, it is necessary to use this framework of quantum physics. And there's an increasing popular field, quantum biology, that is summarizing all the different aspects that needs to be explained with quantum physics and biology. However, I think the quantum physics has many fundamental problems and errors and will be replaced sooner or later with a new framework. This is a bold statement, but as a physicist, I came to this conclusion. So there are many problems that we have now for almost 100 years, and there's, in my feeling, a dead end with all these concepts. And my conclusion is based on published work, mainly from Professor Chandrasekhar Rashtrudri, who is also holding conferences about the nature of light, so only to this specific topic. And this is really interesting to see how difficult it is to interpret the experimental results and to explain it with the classical quantum mechanical framework. Then there's work from Professor Werner Hofer. He could show that the entanglement can be explained nicely with classical physics. Then we have stochastic electrodynamics and Weber's electrodynamics, which also are able to explain many of the quantum physics with the classical framework. So I think this is the way forward, and I want just to have the opportunity to show this here. The third aspect are light effects on brain tissue and function. It has been shown that when stimulating brain tissue with light, there's an increase of glutamate. So there's a correlation between photon emission and neurotransmission. Very interesting. Another thing is the light guiding and light storage property of blood. Blood plasma has the effect to store light for a quite long time. So when you take blood and irradiate the blood, after 10 minutes, still the blood sample is glowing and giving up light. And why is it the case? It has been figured out. The main reason for it is a specific protein, albumin, which has a remarkable property to store light and to give up the light with a very long decay rate. So almost 30 minutes after the initial light emission, there's still photons emitted from albumin. And maybe this is not an accident, but has physiological functions. So maybe our arterioles and veins are somehow also light channels. Albumin is very interesting in the field of medicine because it has been shown that albumin decays in the concentration with age. So maybe this is also linked to the photon propagation in the tissue. If the concentration of albumin is low, then all this phototransmission is not happening that well. Just a hypothesis. There are other structures that are light guiding. For example, the cerebrospinal fluid, where our brain is swimming, has a remarkable property to guide light and to let light travel along these structures. Light exposure and brain activity is also a field that has been investigated and that we are interested. We are constantly exposed to light with different light color, blue light from the devices and so on. And this also affects our physiology, our cognition, and the effects are caused by processes that we understand and also processes that we not understand. So this is a very interesting research field. Then we can use light to trigger maybe healing reactions in the brain tissue. The most famous researchers in the field are Professor Hamblin and Professor Caru, and we are currently investigating if we can couple near-infrared spectroscopy and phototherapy to transcranial near-infrared phototherapy and spectroscopy to apply light and to use also the light to measure the oxygenation. I think it would be a nice technology. Regarding the effects of light on tissue, this is also a very complicated issue and field. There are at least a few things that are quite obvious. There's an effect on the mitochondria and on the cell membranes, shown by many, many studies. And there's a third thing, the effect of water, which is nicely shown by the experiments by Professor Pollack and Professor Zenkova. So, in conclusion, light can be used to measure brain activity and the state of the brain. Light is produced by cells, also in the brain. The light might have biological functions. Light can be used to influence brain activity and neurobiological processes. And neurophotonics is a fascinating and important research area. Thank you very much.
Thank you so much, Felix, for this great talk. And who could be a better choice to do the final plenary presentation at this conference than my co-organizer and colleague over many years, Stuart Hameroff, director of the Center for Consciousness Studies at the University of Arizona at Tucson. Please welcome with me Stuart Hameroff.
Thank you. Thank you, Harold. Thank you all for being here. It's great to wrap up the conference. I want to thank the AV people. Let's see if this is going. Okay, the title of my talk, Do Consciousness, Anesthetic Action, and EEG all derive from quantum vibrations in microtubules? So the rationale and kind of a backstory on this is that 5 theories of consciousness, global neuronal workspace, integrated information theory, higher order theory, OR, the theory that I'm associated with Roger Penrose on, and predictive coding are engaged in an adversarial collaboration sponsored by Templeton World Charity Foundation. The plan is to design experiments to test and distinguish between these theories, narrow the field, and he pushed that very confidently and went away. Okay, thank you. Good job. To test and distinguish between these theories, among these theories, narrow the field and find the most correct theory. And really push, this is a great shot in the arm for this field of consciousness studies. Global neuronal workspace, higher order theory, and predictive coding are more or less cognitive architectures based on neurons and neuronal synaptic networks. IIT ascribes consciousness to complex integrated information states, for example, among brain neurons. Our theory, OR, considers consciousness to derive from entangled quantum vibrations in microtubules inside brain neurons, a deeper level inside neurons. How to test and decide? Well, since we can't measure consciousness directly, one way is several ways to closely examine known neural correlates of consciousness, NCCs. For example, as we heard very nicely and elegantly from George, the actions of anesthetic and also psychedelic drug molecules that we've heard the last few days, which affect consciousness. We can understand exactly how they work. Anesthetic gases selectively block consciousness-sparing non-conscious brain activities, and psychedelics enhance and distort consciousness. OR suggests anesthetics dampen and psychedelics promote quantum vibrations in microtubules inside neurons. The second would be the nature of EEG, rhythms, coherence, and overall meaning, which remain unknown despite nearly a century of use. Specifically, gamma synchrony EEG, 30 to 90 hertz, often known as 40 hertz, is the best measurable neural correlate. OR suggests that gamma synchrony and other EEG derive as interference beats or other types of vibrations in microtubules. And finally, the hard problem, which I'll address at the end. Anesthetics. In the 19th century, a group of gases were discovered which, when inhaled, caused euphoria at low concentrations, and at higher concentrations rendered animals and humans unconscious and unresponsive. For example, ether frolics. If you sniff a little, you get high and buzz and dance around. Laughing gas also was used for party purposes. Eventually, they realized that give a little more of ether, for example, and the patient becomes unresponsive. If you don't give too much, so they keep breathing and maintain their airway, they're perfectly fine, and you can operate on them. And the first surgical procedure that George mentioned at Massachusetts General Hospital. I've done this a long time, but I'm not in this picture. The anesthetic gases have varying chemical structures. They include ethers, halogenated hydrocarbons, and the inert gas xenon, for example. They're quite different, and they have a common effect. But they each have a non-polar, common feature, a non-polar surface region, shown here in gray for all these molecules, which binds and dissolves by extremely weak quantum dipole forces, van der Waals London dipole dispersion forces. That's how anesthesia works. They don't form chemical bonds. They float in, form these very weak quantum forces, and as long as they're there, you're under anesthesia. You turn off the vaporizer, they float out, and you wake up. And they dissolve in a specific solubility medium in the brain. For each gas, a particular average concentration at equilibrium rendered all species, from insects to salamanders to cats, horses, dogs, and humans, unresponsive and anesthetized. The same amount for you and I, a flea or an elephant at equilibrium. This concentration became known as the minimal alveolar concentration, or MAC, and was related to 1 over potency. The more you needed, the higher the MAC. The more potent, the less you needed, and the lower the MAC. Scientists sought a common factor which correlated with potency and searched for the proper solubility phase in which anesthetics dissolved in the brain and body. As George mentioned, around the turn of the 20th century, Meyer and Overton found a tremendous correlation over many orders of magnitude for the anesthetic potency, 1 over MAC, with solubility in a particular medium of olive oil, of all things. So the more soluble in olive oil, the more potent was the anesthetic. And this holds for many, many orders of magnitude. And the most potent anesthetic would be on the far right, methoxyfluorine. It has the lowest MAC, about 0.25%. And nitrogen is an anesthetic at almost 100 atmospheres. So obviously not compatible with life unless you're under special conditions. Now, there were a few outliers which were discovered later. These are gases which bind in olive oil and should be anesthetic, but did not cause anesthesia or loss of consciousness. For example, TFMB and F6. These are non-anesthetics. They're soluble in olive oil, but for some reason don't cause anesthesia. If we can figure out why they don't, despite being in the right place, that would be a big clue. Now, the solubility means this. If you take a body or a brain and grind it up, maybe think of somebody you don't like, and you're going to grind up their brain and find out which compartments in their brain dissolve different types of drugs, polar drugs with charges or non-polar drugs like anesthetics. What you find is that the anesthetics bind in a very non-polar. So going up to the left is polar, going down to the right is non-polar. And you can see Bing, which I use to show consciousness, occurring at the lower right in the nonpolar corner. And on the list on the right, you see that this correlates with xylene, toluene, and benzene, pi resonance groups. Benzene, the basis of organic chemistry, is a ring which shares 3 delocalized pi orbital resonance electrons, forming electron clouds conducive to quantum effects. So there's 3 extra electrons, and they form these clouds above and below the ring. This is pi resonance, and again, organic chemistry. These regions support electric and magnetic dipole oscillations, excitons, charge transfer, phonons, fluorescence. They're quantum friendly. Now, other molecules that are quantum friendly are the psychoactive molecules. For example, neurotransmitters, dopamine and serotonin. You can see the gray area representing pi resonance groups. And on the right, the psychedelics, LSD, DMT, and psilocybin, which we've heard about here at this meeting. They all have pi resonance groups. Now, if you take 2 pi resonance groups, for example, just 2 benzene rings, they attract each other because the electron cloud in one repels the electron cloud in the other, and they form like a bar magnet almost, and then they oscillate back and forth in terahertz, 10 to the 12th hertz, which is in the visible region of the spectrum. And Felix mentioned that, and I'll come back to this point, that there are actually these vibrations in the optical region. And being quantum mechanisms, they can also form quantum superposition. And these are the van der Waals induced dipole couplings that cause this oscillation in terahertz. So basically on the left, you see the dipoles oscillating in what would be the conscious state. And on the right, an anesthetic has come in, which forms its own van der Waals forces. That's how they work. And this dampens the dipole oscillations by dipole dispersions, and that's the anesthetized and unconscious. So where in the brain might this occur? Lipid membrane bilayers have a lot of pi resonance inside. Nucleic acids, DNA, and RNA also do. And of course, proteins are composed of amino acids, some of which are amphipathic, having nonpolar pi resonance groups on one end and polar charged tails on the other. As oil and water don't mix, the the amphipathic amino acids self-organize to form what Operin called micelles with oil-like nonpolar interiors and polar water-soluble exteriors, and this is how proteins fold. So on the right, you see a kind of a schematic of a protein with a nonpolar interior and a polar exterior, and this is just 3, it'd be hundreds of these amino acids. Now, one particular example is tubulin, which has 86 pi resonance rings, tryptophan, which Felix mentioned, phenylalanine, and tyrosine. And in the reds, this work was done by Travis Craddock, who's been mentioned several times. And you can see in the red spheres the sites of anesthetic binding. So the anesthetics are right in the middle of these pi resonance groups in the nonpolar interior of the protein. So I'm assuming it's protein, but I'm suggesting it's tubulin, but it could be others, and we'll come back to that. So anesthetics bind non-specifically by weak quantum forces in fat stores, membrane lipid bilayers, proteins, and nucleic acids all over the brain and body. There's a lot of anesthetic in your body when you're under anesthesia. Yet they selectively prevent consciousness and affect very little else. This is very strange. I often tell my residents there's more anesthetic in your patient's butt than in their brain, but the anesthetic's working in the brain. So in the brain, there's more in the lipid bilayer, but that doesn't mean it works there. This suggests that consciousness involves collective interactions of quantum activities in biomolecules necessary for consciousness. What might they be? For most of the 20th century, it was assumed that neuronal membranes were lipids, which they're mostly lipids. They didn't know about the proteins, and anesthetics were assumed to act by expanding lipid membranes. That turned out not to be true. Jim Trudell and others tried to maintain the idea that anesthetics act in lipids by disordering them, extrinsically impairing membrane protein and dynamics, shown here, preventing a sodium channel from opening. That turned out not to be the case because slight temperature changes cause the same disordering but do not cause loss of consciousness. Finally, Nick Franks and Bill Lieb in the 1980s showed that anesthetics can act directly in proteins in nonpolar hydrophobic pockets, for example, in the membrane-free firefly luciferase. And these hydrophobic pockets are composed of pi resonance groups and aromatic amino acids, phenylalanine, tyrosine, and tryptophan. It was then assumed that anesthetics act in membrane protein ligand-gated ion channels as the presumed site of anesthetic action, and they do indeed bind to receptors for serotonin, glycine, acetylcholine, GABA, and GABA-A, and others. But experiments produced a mixed bag of conflicting results. Some anesthetics open a channel, others close it. Not all anesthetics bind to any one receptor. Leading anesthetic researchers concluded that 2 decades of focused investigations have not identified a ligand or voltage-gated channel sufficient to mediate immobility, and most putative targets provide minimal or no mediation. Opioid, 5-HT, GABA-A, and glutamate receptors and potassium-calcium channels appear to be irrelevant or play minor roles, and no combination of actions on ligand or voltage-gated channels seems sufficient. Well, what to do next? It was assumed that anesthetics and consciousness occur due to membrane protein interactions. There's Bing. What about the cytoskeleton? What about microtubules? As George mentioned, there's a long history of that, and Claude Bernard had shown that cytoplasmic streaming in a single-celled slime mold, amoeba, was reversibly inhibited by chloroform gas. And in the 30s, it was shown that this effect was directly on the cytoskeleton, the cytoplasm, not via membrane signals. And as George also mentioned, Allison and Nunn showed that anesthetics cause depolymerization of microtubules in this echinospherum organism, these axonemes, which were full of microtubules. But this occurred at about 5 Mach, 5 times the amount required. They had suggested in Lancet that anesthesia was caused by depolymerization of microtubules, which, by the way, is associated with POCD, postoperative cognitive dysfunction, that Felix mentioned. So you don't want to depolymerize them. But it did show that anesthetics bind to microtubules. A little over 10 years ago, Eckenhoff's lab began a systematic study of anesthetic binding to all proteins and found about 70 proteins in a neuron, half in the membrane and half in the cytoplasm, that bound tubulin, that found anesthetics. And 3 proteins were, they looked at a couple anesthetics and found 3 proteins common to both, tubulin and microtubules, a heat shock protein, and some other enzyme that don't have any, the latter 2 don't have anything to do with signaling. So it looked like proteomics and genomics pointed to microtubules as the mechanism of anesthesia. They did another study showing, in tadpoles, showing that the fluorescent anesthetic anthracene bound to microtubules and caused the tadpole to go belly up when illuminated, and the anesthetic became active. So in the paper that I think Felix mentioned, and maybe George did too, headed by Travis Craddock and his group in Nature Scientific Reports last year, we simulated quantum dipole oscillations or couplings among all 86 pi resonance clouds in tubulin on a very large computer. What we found was that at KT, at ambient temperature, a spectrum of collective dipole oscillations with the common mode peak at about 613 terahertz in the blue light region of the spectrum resulted. And this was thought to remain internal of the system rather than being radiated. We don't appear blue, although it's interesting that the Hindu deities often appear blue, so maybe they're generating more. But these are in the optical range. We then added each of 8 different anesthetic gases in the simulation in the computer, and each of the 8 abolished the 613 terahertz peak and dampened the other terahertz frequencies proportional to anesthetic potency, to 1 over Mach. So they push the curve downward slower, and EEG also gets slower under anesthesia. We also tested the effects of TFMB and F6. I mentioned the non-anesthetics, and they had no such effect. They did not affect the 613 peak, and they didn't shift the others. Why was that? We then measured the polarizability of the 8 anesthetics and the 2 non-anesthetics, and found that the non-anesthetics had significantly higher polarizability than did the anesthetics. Higher polarizability. What did this mean? We concluded the following. If you look at the top, you see a pair of benzene pi resonance groups oscillating in terahertz. In the middle, you see non-anesthetics on top, round, and because they're highly polarizable, they're going along for the ride with the oscillations. They're not dampening it. At the bottom, you see an anesthetic which has a lower polarizability, essentially exerting some kind of drag on the dipole oscillation, dampening the oscillation and causing anesthesia. So how would terahertz vibrations in microtubules affect what we do? Cognition, consciousness, which is milliseconds, hundreds of milliseconds, much, much slower. Well, we think that there's a multi-scale hierarchy that starts, here's the pi resonance, the terahertz oscillations with the anesthetic effect in the terahertz, and that there are slower oscillations in gigahertz, megahertz, and kilohertz up into the even hertz, into the EEG range. And that this is a multi-scale hierarchy in the brain leading to consciousness, and that the brain, rather than being a computer, is more like an orchestra, more like a multi-scale resonance system. And consciousness may be more like music than it is a computation, metaphorically at least. Not necessarily just sound, but interference, resonance, harmonics, and that sort of thing. There is evidence for this multi-scale hierarchy from the work of Anirban Bandyopadhyay, who looked at neurons, then individual microtubules, and then tubulins at 3 different scales with different types of nanotechnology, and found these triplet peaks in terahertz, gigahertz, megahertz, and then again, gigahertz, megahertz, and kilohertz at the higher level, megahertz, kilohertz, and even hertz. So there is evidence for this multi-scale hierarchy, which we think in the ORC-OR model mediates consciousness, and consciousness can actually occur by Rogers mechanism, which I'll get to at various levels of this, perhaps moving up and down. So conclusion 1 is that anesthetics prevent consciousness by dampening quantum vibrations in microtubules. Conclusion, or conjecture 1A, if you will, is that psychedelics binding to, for example, serotonin G receptors, which are G protein coupled, which means they connect mechanically down into cytoskeletal and cytoplasmic structures, may enhance microtubule vibrations, increasing quantum non-locality and perhaps accounting for the psychedelic effects. This could be via the G protein coupled mechanism, or perhaps by direct entanglement. In fact, there may be entanglement among all proteins with Meyer-Overton binding in the brain. The next topic is the origin of EEG. Now, EEG measures electrical potentials in frequency bands less than 100 hertz, predominantly generated by pyramidal neuron apical dendrites. Gamma synchrony around 40 hertz is the best neural correlate of consciousness. The nature of these frequency bands and their significance remain unknown. EEG has been around for almost 100 years, but when it comes right down to it, we don't know what it really means. We don't know its origin. I read Busaki's book, The Rhythms of the Brain, great book. This rhythm comes from this circuit, this one comes from that, this is a pacemaker. Gamma, we don't know. In the end, it doesn't hang together. It's still a mystery. So where might EEG come from? So the work of Arad Kalra in Jack Tosinski's lab, and Arad gave an excellent presentation the other day, it points out that microtubules play a bioelectrical role in the cell, that ions flow along and through microtubules. So here's ions. Microtubules are negatively charged, and positive ions will attract and move along them and actually a current flow, and actually also go into the lumen, kind of like a membrane ion channel at a smaller scale. Now, Cantiello's lab, Horacio Cantiello, who was at Harvard, is now in Argentina, used patch clamp techniques on microtubule bundles and tubulin sheets, and they found that tubulin sheets and microtubules emit voltage-dependent electrical oscillation with a peak at 39 hertz. That's pretty darn close to 40 hertz oscillation gamma synchrony, which isn't an exact number anyway. Arad's paper also found a flip in resistance between 10 and 100 hertz, roughly the EEG frequency range, and and increased microtubule conductance with a peak at about 40 hertz. So there's some evidence that EEG might come from vibrations in microtubules either as a beat frequency, interference beat from faster, or directly, or some combination. So conclusion 2 is that microtubule oscillations are the likely source of EEG rhythms, including gamma synchrony, the best neural correlate of consciousness. Finally, I want to address the hard problem, particularly what Dave had to say earlier. The nature of phenomenal experience composed of qualia remains unknown, the hard problem. Penrose proposed objective reduction, OR, a self-collapse of the quantum wave function, produced consciousness. The wave function describes superposition states, particles in 2 or more locations or states simultaneously. Why do we only see definite states? We don't see superpositions. Roger began by essentially telling us what superposition might be. Despite all these years, nobody's ever explained how something could be in 2 states or locations at the same time. Roger began by portraying 4-dimensional space-time as 2-dimensional space-time sheets, shown here. From Einstein's general relativity, he represented particle locations as specific curvatures in space-time locations. So this particle is oscillating back and forth, as is the curvature that defines it. So space-time curvature is oscillating back and forth. And being quantum, these can also be in superposition of both curvatures at the same time. Superposition is then portrayed as a separation in space-time geometry. Now, you might imagine that were such separations to continue and there wasn't any collapse, you'd get multiple worlds. Each possibility would branch off and form its own universe. And as silly as this seems, it's a very popular belief among physicists, as is what Dave was saying. Silly. According to the Copenhagen interpretation, which is what Dave was talking about, consciousness collapses the wave function and causes quantum state reduction, or what I call subjective reduction. So here's your conscious observer over here, Bing, looking at this superposition of space-time sheets, causing this one to stop, cease to exist, and this one to continue. So this is collapse of the wave function. It doesn't explain what's going on in the brain to cause consciousness. It puts consciousness outside science. It's dualist, puts consciousness outside science. Now, Roger said that these superpositions, separations, are unstable and will self-collapse, that is, undergo objective reduction at time t equals h over E sub g. E sub g is the gravitational energy required to separate the space-time or a mass from itself, along with a moment of consciousness. So he said, when this happens, bing, there's a moment of consciousness happening due to the self-collapse. So in other words, rather than consciousness causing collapse, as the Copenhagen interpretation and subjective reduction would be saying, collapse OR causes consciousness or is consciousness. Bing. So this would be happening down at the very low down level. So just to reiterate, the Copenhagen interpretation, consciousness causes collapse. Penrose objective reduction, collapse causes or is consciousness. Now these events would be happening in the environment all around us, in the lectern and the air and so forth, and they'd be random and lack meaning and memory and so forth, and would be what we call proto-conscious. And they would be similar to, they would be random and similar to what Whitehead called simple occasions of experience, or what might be viewed as decoherence. What's called decoherence might actually have proto-conscious moments going on all the time. So the question is, how do you combine these into something full, like full, rich, conscious experience? Metaphorically, I think of these proto-conscious moments as like the notes, tones, and sounds of an orchestra warming up. Noise or a cacophony. If you go to the symphony and they're warming up and each musician's tuning his or her instrument, you hear these sounds, notes, tones, noises. It's not music, it's annoying, it's a cacophony. And then they play Brahms or Beethoven or the Beatles or whatever. So how could proto-conscious moments be organized or orchestrated in the brain for full, rich, conscious experience? I think this is essentially the combination problem in panpsychism. And this has a tremendous advantage over panpsychism, which, as far as I can tell, is a property of matter. And OR is what is selecting the state of matter, so it precedes that. So it's like the combination problem. So basically, we're suggesting some kind of isomorphism between superpositions and collapses at the level of space-time with what's going on in microtubules. So the answer is to orchestrate and unify by quantum resonant vibrations these quantum processes in tubulin and microtubules. Now, a couple other things. I've suggested in another paper that I don't have time to discuss here that the origin of life in the primordial soup was due to these amphipathic pi resonance groups coalescing and forming clusters, which then would collapse, some of which would give rise to pleasure, primitive pleasure, or quantum pleasure, as I've called it, which would provide a feedback fitness function for the development of living systems and drives evolution. I think we have to include pleasure and feelings in evolution. But to do that, we have to have a mechanism to produce feelings and pleasure in biology, and this may be it. So the pi resonance groups have only a few stable arrangements, and S may be metastable, so there may only be two stable arrangements, and it could be that one of these arrangements is displeasurable and one is pleasurable, and this would provide the feedback fitness mechanism to drive evolution to seek pleasure, not just hedonism, but altruism and spiritual pleasure and what have you. So since apparently a big selling point of Tononi's IIT is that it generates some kind of patterns that seem to map somehow with qualia, I'd like to point out that the qualia space in tubulin microtubules shown here, and I'm proud to have introduced emojis into consciousness studies a few years ago. And you can see the possibility with 86 interacting with the other 86, the possibilities are enormous for the number of qualia and configurations of qualia and feelings and conscious content in one tubulin. And then you have about 1 billion tubulins per neuron, then you have 100 billion neurons. So just to summarize, let me say that consciousness and anesthesia and EEG doesn't come necessarily from the membrane, but from what's happening inside the neuron. Here's a pyramidal cell in the microtubules going down to these pi resonance groups. So in conclusion, let me say the following. Unlike other theories of consciousness, OR can account for the mechanism of anesthesia and psychedelics. Unlike other theories of consciousness, OR can account for the origin of EEG. OR has a viable approach to the hard problem through Penrose objective reduction. And finally, OR can in principle account for non-local effects of consciousness, something that was discussed yesterday, I believe, the origin of life and evolution driven by pleasure. Thank you very much.
Thanks very much, Stuart, for this great talk. And may I ask you to take one of the seats?
Okay.
May I also ask the other speakers to come
water?
up on stage? Sorry?
Water.
Some water.
Yeah.
Yes. And can someone bring 3 bottles of water from the cooler down there? That would be nice. Now we are in the Q&A period for these 3 talks. And I would like to ask you to line up behind the microphones for questions. Let's just start with the first question right in front. First question to you.
Thank you. I have a question for Stuart Hameroff. Stuart, I love your talk. Has it ever struck you that critical point fluctuations could play the role of the oscillations you have?
Yes.
And that you could achieve the same kind of interference with critical point oscillations that you're talking about flipping between 2 states. That would link my theory with your theory extremely closely. Thank you.
Yeah, thank you, Alex. I have considered that, and critical fluctuations, including proposed by Kaufman and the poised realm and all that, I think is very relevant. But what is needed is a medium in which this is happening that would be relevant to consciousness. So if you take what you're doing and what Kaufman's doing and what other people are doing and apply it to microtubules, I think that'd be a winner.
Thank you so much.
The gentleman in the back?
Myself?
Yes.
Okay.
Are you a gentleman? Yes.
I hope so. Just to clarify what you mean by non-local effects of consciousness and how OR contributes to that. If you just make a clarification exactly what you mean, because some people are talking about non-local in all kinds of ontological ways.
I'm afraid that's what I mean. I mean, I don't think, people talk about near-death, out-of-body experiences,
Yes.
Reincarnation, afterlife, and so forth. I'm not a big proponent. I don't advocate it. I don't argue for it. I don't try to study it. But I don't think we can rule it out until we know. We can't exclude consciousness out of the brain until we can explain consciousness in the brain. Okay. If we completely explain consciousness in the brain by classical effects, then all the other stuff is impossible. But if it's a quantum effect, there's non-locality, then who knows? I mean, parapsychology, all that stuff. I think there's a fair amount of evidence for it, and it's not something I want to dwell on, but I don't think you can rule it out.
Thank you.
Next question, please.
A question to the anaesthetists on the panel as a fellow anaesthetist. I remember Antonini did some work in the 90s looking at the site of action of MAC, or demonstrating that it was predominantly a spinal effect. So how do you rationalize your data with the fact that MAC is significantly lower if you apply it to the spinal cord?
Well, that's not what he said, but you want to answer that? So Antonini, a bunch of people did that. I actually reviewed this recently. So what they did was they measured MAC in an animal. They did it in a couple different animals. He wound up working on goats, I think.
They did, yeah.
And they wanted to find out where anesthetic acted and where consciousness acted. And let me preface that by saying that prior to that, the powers that be in the anesthetic mechanism research world, namely Ted Eager at UCSF and Keith Miller at Harvard, had decided that the definition of anesthesia, loss of consciousness, amnesia, and immobility, they don't move when the knife comes, was too complicated because, as they said, we don't know what consciousness is. Dan Dent and other people say it doesn't really exist. Let's just get rid of that and define anesthesia as immobility and amnesia. So that's the context in the 80s. So what Antonini and others did was, okay, let's take an animal and we'll start from the top. We'll decorticate them and measure MAC. We'll measure MAC in the whole animal, sever the cortex and measure MAC, and it was the same. Okay, let's go a little bit deeper, down to the subcortical, cut that. Okay, same. Cut the thalamus, da-da-da. They finally got to the spinal cord and found, but what they were measuring was movement. They weren't measuring consciousness. They were measuring movement, and they weren't measuring memory. So what they determined was that there was no movement if the spinal, or that immobility, not loss of consciousness, not memory, was mediated by the spinal cord. You're an anesthetist, so you know that patients can move and not be conscious and certainly not remember. And it's somewhat of a controversial point, but they didn't show anything to do with consciousness, and it was in the context of getting rid of consciousness from anesthesia, and George and I and a few others are trying to bring it back. So it just tells us that it's like a spinal reflex.
Yeah, it's basically just saying that MAC, which
MAC, which is intrinsically linked to movement by definition, is mediated at the spinal cord. It doesn't suggest that anesthetics don't work in the brain to cause loss of consciousness.
I entirely go along with that MAC is just a measurement of immobility, so it doesn't tell you whether the patient's asleep.
Right, they don't move.
That's right.
That's all you know.
Okay, thank you. The next question goes to the back.
A question for Stuart. I wonder how ORCOR address regular sleep and maybe circadian biology.
Yeah, that's a good question. I don't really know. I do, maybe George probably knows more about this than I do. But I will say that a decade ago, where people ran into brick walls trying to figure out how anesthesia worked, switched over to, well, it's just augmented sleep. All anesthesia is doing is augmenting normal sleep mechanisms. And I don't think that's true. So other than that, I'll pass to George. Do you have something to say?
Yeah, well, first of all, I'm not going to suggest that OR explains normal sleep-wake states, nor anesthesia, nor consciousness. I like Stuart, and I respect his strong opinions, but I do want to be clear for this audience. Stuart speaks with great authority, but he is the only person who would get up and attribute all of the actions of anesthetics or consciousness to microtubules, and I think you'd agree with me on that, first of all. And second of all, I think the relevance of 40 hertz oscillations, I completely disagree that is the best neural correlate of consciousness. Gamma synchrony can be observed during sleep. It can be observed during anesthesia. In fact, it can be enhanced during these unconscious states. Higher gamma frequencies, which are thought to correlate with broadband neural spike activity, might be better correlates, but I do not think that the 40 hertz is the best correlate. So I am more of a traditional neuroscientist, and I look to the sleep promoting regions like the ventrolateral preoptic nucleus and the median preoptic nucleus and the hypothalamus as generators of sleep and for mechanisms of sleep homeostasis.
Thanks.
Thanks very much, George. The next question is here.
A question for Stuart. If consciousness doesn't collapse the wave function, how do you account for free will? And how did your theory not become epiphenomenal?
Yeah, good question. First of all, how do other theories not become epiphenomenal? But let me address, I think free will, there's a lot of issues with free will. I wrote a paper called How Quantum Brain Biology Can Rescue Conscious Free Will. The first problem you run into with free will is that if you and I are talking at a cocktail party or something, and you say something and I answered back immediately, if you measured the activity in my brain corresponding with what you said, I would have already answered you. And therefore, the powers that be in neuroscience philosophy say that I answered autonomously or my non-consciously, robotically or something, my inner zombie, and then subsequently had a false illusion of answering you consciously. That's the party line. That started with Dan Dennett, who completely misinterpreted Libet's experiments, and they continue to be misinterpreted. Now, how do you get around that? Now, you get around that by temporal non-locality. That's something that Harold's very familiar with. That would appear from the classical world to be backward time effects. And if you go back to Libet's sensory experiments, where he had exposed brain in awake patients and would stimulate the finger and record from the finger area of the cortex, or stimulate the cortex and ask the patient when they felt it, they find that if you stimulate the finger, you get an evoked potential and an almost immediate response. If you stimulate the cortex, it takes 500 milliseconds of activity, which is a long time, a half a second, and then you feel it. If you try to trick the system and stimulate the thalamus, you get an evoked potential, but if you then stop the activity at less than 500 milliseconds, they don't feel it. He concluded that somehow the brain or consciousness or whatever knew what was happening, what was going to happen for the next 500 milliseconds. If you didn't fill out and get that 500 milliseconds of activity to reach what Libet called neuronal adequacy, there was no conscious experience at time 30 milliseconds or 90 milliseconds, whatever the evoked potential was. Therefore, at the time of the evoked potential, the brain has to know what's going to happen 500 milliseconds subsequently. And his answer was backward time effects, which is okay in quantum physics and time symmetry. So you need that. Now, with that, then I could be answering you consciously, despite the fact that my brain activity that seems to correlate with what you said hasn't happened yet. People have said the same thing about baseball or cricket players hitting a really fast pitch, or even tennis players, that you need to know in advance where it's going to go, what's going to happen in the near future. Now, having said that, even if that's true, you still have the problem of determinism. Maybe, okay, I acted in real time consciously, but maybe I was predestined to do that because of everything that went before and after. That's a separate problem. But I think just to have the possibility of free will, the possibility of acting consciously in real time, you need temporal non-locality.
Next question, please.
Thank you. There are two questions for Dr. Felix. The first is, have people studied the quantum nature of the ultra-weak photonic emission from the human body? Because that would give us an understanding of the quantum nature and quantum effect, whether it involves quantum effects like coherence, etc. The other question is that you didn't mention optogenetics in context to light in brain. So what do you think would be the implications of optogenetics for consciousness research? Thank you.
Thank you for the questions. Concerning the first question, this is an ongoing study to see if the light that is emitted has non-classical properties. And there are some preliminary results indicating that it's maybe squeezed light, so it's a squeezed state. However, the interpretation of the result is not necessarily true, I would say. And the proper experiment that needs to be done is a classical double-slit experiment and all these quantum physics experiments with a light source being tissue. This has not been done, unfortunately, but needs to be done, of course. The second question about optogenetics, yes, this is also part, of course, of neurophotonics. So that needs to be also included in this talk in principle, but here I was only referring to human experiments, and optogenetics in humans is still not been done. There might be a role to investigate this also safely, and this is part of the future research, yes.
Thank you.
Thank you. Next question.
I wonder if the quantum effects also have a role with the intravenous anesthetics. Like I'm thinking, like propofol, it dampens the electrical activity, and on the other hand, like ketamine may have opposite effects, like classic psychedelics, the dissociative state. And also, do you think ketamine would it be able to shut down like the 24/7 awareness that some experienced meditators describe, and that is shut down by anesthetics? I don't know which ones.
Propofol and ketamine get inside neurons and bind to microtubules. There was a talk the other night about propofol and microtubules. If you look at propofol, it's got a aromatic ring and then a couple of polar things, which make it soluble. And I don't know if those things break off for it to get inside the neuron or just gets in. But it seems to get inside the neuron. Now, it also binds to GABA receptors and whatnot. So, and it could be that drugs can bind to membrane receptors that secondarily affect the microtubules, and the microtubules could still be responsible. But I have seen images and studies showing that ketamine binds to microtubules not just in microtubule preps, but in cells. So they do. In fact, babies who get repeated ketamine. Babies and old people are susceptible to POCD, postoperative cognitive dysfunction, if they get a series of anesthetics. And we've gone to not just giving them gas or propofol, but switching it up in the hope that would minimize the postoperative cognitive dysfunction. But it doesn't seem to, although this is a controversial area. Maybe George can comment on it. But it seems to me that if there's a unitary action of anesthetics and they all work the same, then whatever anesthetic you give is going to have similar toxicity because it's going to be binding to the same target. And if it's microtubules, it's going to be destabilizing the microtubules, as we know that 5-MAC can do, going back to the Allison and Nunn experiments. So I think, I don't know about etomidate, but ketamine and propofol do get inside the neuron, I think.
So
That might be true. I don't know of robust evidence related to ketamine working in microtubules, but I do want to remind the audience that propofol and ketamine also bind to well-described ligand-gated neurotransmitter receptors as well as various channels. And for example, HCN1 channels, which are thought to be have a pacemaker function in the brain, are thought to be important for ketamine's effects. When you knock those out, the hypnotic effects of ketamine are attenuated. NMDA receptors, another target. So again, I don't want to seem like I'm in a state of quantum displeasure, but I really want to ground the audience that for every comment that I'm hearing about microtubules, there are about 1,000 papers testing other traditional targets at the cell membrane.
And what have they come up with?
Well, they've come up with quite a bit, in fact.
Well,
so, I mean, the fact that you could, for example, alter HCN1 expression and alter the hypnotic effects of ketamine, or deplete, for example, getting to a circuit level, noradrenergic neurons and the locus ceruleus and find out that you potentiate anesthetic effects. I mean, there are an array of emerging data related to not just the molecular level, but the neural circuit level that can account for hypnotic actions in a way that's consistent with other states of unconsciousness, such as sleep.
I don't want to get in a fight with my friend George. He's one of the few friends I have in the anesthetic mechanism world. But let me just say that, the anesthetic gases all bind to these. They all bind to GABA. They don't all bind, but many of them bind to GABA. Some open it, some close it. Not all bind. It's the same with other receptors. And I'm sure that, and it could be true of propofol, which binds to GABA, but may exert its action inside the neuron. People always say, well, if you do a point mutation in the GABA receptor, it changes MAC. Well, a lot of things change. MAC is the potency of it. A lot of things change MAC. Age, weight, various metabolic conditions. So that's not really the answer. So I don't think. But the point is there are thousands of papers out there. And yet, the paper I went over quickly by Ted Eager et al. In 2008, who was the leading proponent of membrane receptors as the target of anesthetic action, basically gave up. He said it's impossible, and I read those quotes.
But wait a second. To be clear, and I saw the quote up there, impossible with respect to immobility. The other thing that we need to point out is that there doesn't necessarily need to be a unitary hypothesis. Anesthetics result in a similar functional outcome, which is we can take care of a patient, we can intubate them, they're not responsive. The states that they induce are not the same. If the percentage, for example, of dreaming during a ketamine-based anesthetic is 80 to 90%, as opposed to 10 to 20% for something like propofol or desflurane. So they don't all work the same, they achieve a similar functional outcome. And one other point I need to dispute. I'm your only friend in anesthetic.
Not one of them? No, that's not driving the other friends.
I think we're seeing here the continuation of an adversarial collaboration.
Yes.
The next question goes to the back again.
Hi, hello. So this is a question about the biophoton emission. If organisms are constantly giving it off and at the same time receiving it, do you think this is a method of communication between the environment, between organisms? And if you do, what kind of information do you think?
Well, this is a good question, and it has been also investigated. There are many different studies performed using single cells, colonies, or single cell organisms like Paramecium, if they communicate even when they are in two different cuvettes and there's a light shielding, for example, or if there's a light channel between them. And the experiments indicate quite clearly that there is some communication happening between organisms via optical communication. So I think this is quite evidenced. What is happening actually if there's a modulation of the signals and so on, this is not investigated. mainly because it's so difficult to measure it. I mean, the photon emission is so weak. We speak from of 10 to 100 or 1,000 of photons per second. So you need really photomultiplier tubes to measure it, and then all these the discrete states are making it very difficult to analyze the data and so on. Yeah.
Thanks, Felix. Petra, this is your next.
Yeah, still, even though it's not easy to measure all these things and they're so subtle in the way they occur, I would like to, mirror my perspective on this question of light. I do think in our culture, with the development of the mind and the way we mirror it or symbolize it culturally, we have this symbolism of light, like the spirit and the mind being light, and maybe the body being dark matter, being black. And I do think psychologically we can get in touch with all these, the spectra of the light from, not being, it's not on the physical level, but it's on the psychological level that we see that certain patients are really in the darkness. And maybe it has to do, if you look at spiritual experiences, people are really, very stuck by the thought or the impressions of light inside their bodies. I've heard about the light going up the spinal cord or the light surrounding the entire body. And definitely nobody, of course, is seeing this. It's an inner process or inner processes taking place. And in some ways, I do think it's actually a proof, or not a proof, or an example, or some way to see that there's actually this connection between the psychological, the spiritual, and of course the physical measured realms, as you now are working on the light. And I'm just very intrigued by this. One could talk more about that, but how do you think about this?
Thanks for the comment. Mm-hmm. Of course, in all that states of consciousness, you can experience light. I have also the phenomena experienced by myself. If this is related to ultraviolet photon emission or not, this is another thing. But of course, there are visual impressions that can be stimulated by different techniques that are maybe related to these phenomena.
Yeah. I'm just thinking with it in the terms of autopoiesis, that mind is really reflecting matter and vice versa. And so, well, but thank you.
Next.
Great talk by all the speakers. Thank you very much. My question is for George Mashour. It's a three-part question. One, is that work published yet?
It's under review.
Okay. Two, have you thought about doing the same thing with other chromophores such as tryptophan or any of the other ones that Dr. Scholkmann mentioned?
I haven't thought about that. I mean, this was sort of the initial proof of principle. Can we do this technically? Can we introduce an anesthetic? I guess what I've thought about is how to, as I mentioned at the end, how to make it more biologically relevant by, for example, introducing synaptosomes or there are cortical slice models that you can pass photons through, so something more biological. I'm not sure if my colleague, Dr. Goodson, has looked at this because, again, he's really in the business of studying the energetics of chromophores, both natural and artificial.
And the final thing, are you familiar with the work of Robert Alfano out of City College, where he's sent light into brain tissue and measured the entanglement as it comes out?
Yes, that's just what I meant in terms of passing it through a cortical slice, and that's another possibility that I've explored for that experimental setup.
Okay, excellent, thank you.
Thank you.
Jürgen's next.
So I also have to thank to all for the fascinating session. This is a question to Stuart. Microtubuli are widely distributed in the world. Do you think, according to your theory, we will be able to decide which species or which animal has consciousness and which not?
Well, first let me say that according to OR, there's proto-consciousness happening everywhere. And according to panpsychism, there's some kind of consciousness everywhere. So the question is, does it get organized sufficiently to be the type of consciousness that we should care about or worry about in terms of treating an entity. You can also look at it, so in the brain, so there's microtubules in all the cells, and plants, by the way, and which is why I made that point the other day, that the cognition in plants could be mediated by microtubules. The issue is collapse occurs, according to Rogers' equation, by E, which is the amount of superposition, equals H over T. So it's inversely proportional to time. So if you have a single cell in your skin or an amoeba or something like that, and all its microtubules are coherent, it might have a conscious moment every couple minutes. But before that happens, something might disturb it. We have so many microtubules aligned in anti-parallel in the dendrites and cell bodies of pyramidal cells and throughout the brain, billions and trillions. Actually, there's 10 to the 19th tubulins in the brain. That if they are entangled and coherent, they can reach threshold and collapse in terahertz, for example, or let's just say megahertz, a million times a second. So that means we could have 1 million conscious moments per second, and then they interfere to give interference speeds that come down into the EEG, because we don't think that fast, although our subconscious might, we don't know. So you need the specific arrangement in the brain to have the kind of microtubules, particularly the anti-parallel microtubules, so that they have slightly different frequencies in a common voltage. So they're going to give rise to interference speeds, which is going to give cascades of resonance down into the slower region, including EEG. So I think the brain is set up to have the type of consciousness we have. As far as organisms, I think any organism has a chance to be conscious if you leave it alone. If it has enough time to reach threshold, and the intensity of the experience might be related to inversely the time. So it might be an electron in superposition would reach collapse and have a proto-conscious moment in 10,000 years. But it would be pretty boring, I think, because it would be very low intensity. So we have a lot of them of high intensity thanks to our brain.
And could I also just point out, too, that in addition to microtubules, single-cell organisms and plants also have ion channels at the membrane, and they have excitable membranes, and you could detect action potentials, as we heard from Paco and others, in plants as you can in neurons. So there are alternative substrates across species and organisms for generating the kind of electrical activity that might be associated with computation.
Let me just say that's a point well taken, and the Templeton project, because what you just said, ion channel membranes, is represented by the other theories primarily. I mean, they all attribute consciousness and activities to membrane activities. So hopefully we're going to find out over the next few years.
How many years? I see 3 final questions. I would say the first one of the 3 goes back to the back microphone.
This is very short for Stuart. You mentioned frontal gamma brainwaves as the criterion for consciousness. But Jonathan Shear, who you obviously know, and Fred Travis, who you may not, published a paper saying that there are 3 basic kinds of meditation. One keeps you outwardly organized and is called focused attention, which produces frontal gamma. The other is called open monitoring and is a typical kind of Buddhist technique and produces midline theta. But the one which produces what Jonathan Shear called pure consciousness, which he devoted his life to, is a global coherence, not just in alpha, but often in beta 1, beta 2, and gamma as well. So I just offer that thought to you, that if you include pure consciousness, it's the coherence which matters.
I would agree with that, and I said gamma was the best, and George disputed that, and I think you said higher frequencies might even be better. And as I recall, the original studies on meditators done at Davidson's lab, the Dalai Lama sent his best monks to Wisconsin 20 years ago or whatever, and they measured them. They found that their baseline gamma was higher. Instead of 40, as most of us would be, they were in the 80 just before they started, and then when they meditated, it went really higher. And there's probably much faster stuff going on that we can't measure. And the higher the frequency, I think the quote-unquote more conscious you are. And I think probably psychedelics do the same thing. In fact, there's some work on psychedelics from Carhart Harris and others that patients subjects under psychedelics, their MRI and their EEG may actually look cold and dark, at least in large parts of the brain. And their cognition is not very good. You wouldn't want them driving you home, but they're highly conscious because, in my view, consciousness has gone to a deeper level, more into the quantum realm with non-locality.
I just want to add, though, that it's important not to conflate coherence or synchrony with neuronal communication. I mean, there is likely some degree. You certainly need temporal coordination, and there's certainly a range in which you need some kind of coherence. But global coherence, in a highly phase-lock way, could also get you a seizure, which is an unconscious state. Also, I want to point out that there are distinct generators for gamma, and 40 hertz, or that around 40 hertz, is different from a higher gamma bandwidth.
Thank you, George. No. Come on, no. Please take your seat, and the next question goes here.
It was just because you said an electron could sail away and have a moment in 10,000 years, and the thing I've been wondering about is the time scale of when this in the orchestrated OR model. What kind of time scale are we talking about if an electron before it undergoes spontaneous reduction?
Well, you need a lot of them to get more E sub G. So one electron would have a very low E sub G, which is equal to H over T, and the T would be very long. If you have a lot of electrons also pulling on the nuclei, you need the nuclei to get a significant mass. And a 1 nanometer movement of the electron moves the carbon nucleus its diameter, femtometer, which is enough for superposition and gravitational self-energy. So you need, I think it's like billions of tubulins in superposition to have a threshold in a physiological range. And for the 10 million, you need a lot more. Fortunately, there's a lot of tubulins in the brain. So you need a lot of them to be coherent. And let me respond to something George said about coherence not being the same as communication. I agree with that. In fact, there was a recent paper from, I think, of some Carhart Harris's group also, where there was patients in the psychedelic state with high degrees of coherence across the brain, but no Granger causality. So there was no communication of that nature from one part to another, and yet they were highly conscious. And that reminded me of EPR, Einstein-Podolsky Rosen entanglement, where you have separation, you make a measurement here, and this one responds. And that's not causal, and it's not signaling. It's something else, something nobody has a really good name for it. So that could be what's going on in the brain, that there's some kind of quantum correlation without causality that correlates with a highly conscious state.
Yeah, and you don't even need to invoke quantum physics there. I mean, I think my general point was, I mean, if you have no coherence, you're not going to have communication. But if you have nothing but synchronous discharge, and there might not be any information transfer between two regions.
Next question, please, in the back.
I was wondering if you had studied the relation with your series, Orker and others, in relation with memory, like better access to memory, better writing to memory, absence of writing to memory in normal consciousness.
Well, the first question is, where the heck is memory? The idea that memory is stored in synaptic plasticity is the party line, LTP, and so forth. But the synaptic proteins that are synaptic plasticity are only there for hours to days. They're turned over thanks to microtubules and exoplasmic transport, et cetera, et cetera. And we, meaning Travis Craddock, who was a previous questioner, Jack Tuszynski, and I and others, have published a couple papers suggesting that memory is encoded in microtubules, and we showed that CAMK2, which is activated when calcium comes in LTP, transforms into this holoenzyme with hexagonal kinase domains, which exactly map onto the hexagonal lattice in microtubules and can phosphorylate. And we think that's a good mechanism for memory. And the microtubules and dendrites are capped and quite stable. So since neurons don't divide, they don't have to depolymerize and to be used for mitosis. So they're there a long time, if not your whole lifetime, because they're capped at both ends. And so they're an ideal location to encode memory. So I think memories in microtubules would be my answer.
George? Anything to add? No. Thank you. Please.
Some of you might be familiar with my work when I can actually modify brain oscillation by delivering near infrared light and delivering at different frequencies. Now, the point, so we published in scientific reports and how it changes. I don't want to go into details about that, but I think the discussion about gamma and beyond is really interesting. Now, I think gamma is very historical. We've delivered gamma, and we see changes in the high frequencies, reduce the power of the low frequencies. Fascinating. So we can induce changes, but we've gone into meditation and what the effect is, and there is some discussion about higher frequencies. Now, this is very interesting. We've been experimenting, and I've been doing it. You saw maybe Felix was there, but we had a different understanding about how it's done. But there's some meditators here who have experienced it, and when we induce higher frequencies than gamma, long-term meditators have like a window. They hit it, and they go into these altered states. It could be some vibration, it could be the unity with the environment and blocking of awareness and more into your body, etc. So we can discuss, I think some of the people are here. But it does seem that there's more than gamma. So depending on the level of, I don't know what it is, the quality of your, or the sensitivity to different frequencies, I think people and your body and your cells have been oriented or trained to be able to do different things at different frequencies. So that's yet to be explored. We're going to do a meditation study involving advanced meditators and how that is able to get into advanced states. We will learn more, but I just want to say that gamma is great, it's historical, it does actually some physiological effect that's shown by MIT in the Alzheimer's study. That's why we do it for Alzheimer's study. But there will be, actually, we have to be open to higher frequencies and what the effects are. I think we will discover that more. Thank you.
So is there any question associated with that, or is that just a comment, right?
More work.
Okay, thank you. Then there is the gentleman over there, and then we close the questions and answers part of the session.
1935, Einstein published the EPR paper. There was another paper that he published that year also, called the ER paper, the Einstein-Rosen paper, which introduced the notion of an Einstein-Rosen bridge. If you have two massively entangled black holes, the solution from general relativity is that those entangled black holes always be joined by an Einstein-Rosen bridge, which you can look as a shortcut through space-time. 6 years ago, Juan Maldaceno at the Advanced Institute and Lenny Susskind at Stanford published a conjecture that any time you have entanglement, necessarily you have an Einstein-Rosen bridge. Might this solve some of the problems that deal with non-locality, et cetera?
Does any of you want to respond to that?
Yeah, you're suggesting some kind of wormhole, I think. But I mentioned this the other day. I was fortunate enough to be invited by Roger Penrose to the 1998 meeting at the Royal Society in London, which was the first meeting on quantum information. For the number of years prior to that, there had been a whole lot of stuff showing entanglement across distance. And they had just done the experiment between two villages in Italy, Nicholas Giesen, and now they've extended the distance more. But it was And of course, aspects experiments were only 10 years before that. So it's been about 10 years of entanglement being proven rather than theoretical. And all these young physicist guys at the Royal Society meeting were trying to explain it. And they kind of kept hinting at something. And at the end, Roger got up with a blackboard and said, I think a lot of people have been addressed the question of how you can have signaling faster than the speed of light over non-locality, over distance. So he drew the EPR where there's two things here. One goes here, one goes here. You make a measurement here, then this one instantaneously collapses to the complementary. And he said, the answer is obvious, but everybody's been afraid to do it, but I'll say it because I'm the only one who's already a member of the Royal Society. You make a measurement here, and the quantum information goes backward in time to when the two were together, then informs this one. That's the answer.
Okay, thank you, Stuart. We're approaching the end of this session. It's the last plenary session of this conference. And before I really come to its end, I want to, well, those of you who have dinner tickets for tonight, just let you know that the dinner tables will be prepared until 7 o'clock. At 7:15, you can enter the room, and at 7:30, dinner will be served. It will be a seated dinner. There's no buffet, so you can relax, you can sit, you don't have to run around, you can talk to everybody on your table. It's a very European, civilized way to dine. So, that being said, I want to also remind you of the next conference on the science of consciousness, which will take place in Tucson again, April 12th to 18th. Save the dates in your agendas. 12th to 18th, it will again be in the Ventana Canyon Resort. So those of you who have been there before know how beautiful this place is. It doesn't have mountains, but it has saguaros, right? And some time of the year it's cooler there. That's right, that's true. There are some mountains, yes. Not like these.
Wow.
Okay. And so this conference will again be organized by Stuart and by Abby. Abby is not here at Interlaken, but I invite you all to apply and with your abstracts and come to participate in this conference. Now, since this is the last time in this conference, at which we are all together in this room, I want to use the opportunity to thank you all for your patience and perseverance given the weather situation. I hope over the last days the water consumption was satisfied with what was offered. And most of all, I want to thank you all for making this conference such a success. Thank you very much.
Thanks to Harold and Gundel.
Thank you.
All right.
Thank you all. Coffee break, and then you go to the concurrence, and don't forget, 7:15 dinner will start.